This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 1133, recorded on July 19th, 2024. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Fort Lee, New Jersey, Dixon Despommier.
Hello, Vincent, and everybody else. It's an amazing day out today. It should be hotter than you know what, but it's not. The humidity should be higher than you know what, but it's not. My wife and I will have dinner out on our porch tonight. Whatever we have, we will enjoy because we haven't been able to go outside for about a week and a half. It's remarkable. That's all I can say.
Here in New Jersey, it's 29°C, sunny, and 41% humidity. That's like half of what it's normally.
Look at that.
Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. I have to say that it's really good to see everybody. You guys are my friends, and I really enjoy this. This is good fun. I got warmed up because we had like 10 minutes of talking to each other in the pre-show that these guys all miss. But this is great. Okay, so it's 90 degrees and partly cloudy. And I have to say that for Austin in July, that's like weird even, okay? It's not hot. It's not 100. It's not 110. I think last year at this time, last year was awful. We had, many, many days well over 100 degrees. And get this, next week, starting Tuesday, for the ensuing 6 days, the forecast is for less than 90 degrees, as low as 84, okay, with significant possibility of rain. This is great, except that I'm going to be in Maine, where the forecast is even hotter than what it's going to be in Austin. So there you go.
Right.
What's your humidity today?
50%.
It's not bad.
It's not bad.
Not bad at all.
All right. Also joining us from Ann Arbor, Michigan, Kathy Spindler.
Hi, everybody. Here it's 78 Fahrenheit, which is 25 Celsius.
Nice.
Just perfect weather for art fair. It's usually hotter than blazes for art fair. And then it would rain in the late afternoon. All the art fair people have to put their stuff away, and then it dries out and everything's really humid. So this is amazing weather. So, yeah.
And from Western Massachusetts, Alan Dove.
Good to be here, and great to also be a member of the lovely weather club. It's 80 Fahrenheit, 27°C, dew point 56, which is the lowest it's been in weeks. This is the first, I think this is the first really nice summer day that we've had here.
I want to tell you, as a coda to our weather warm up here, that I am in touch via Facebook Messenger with our local meteorologist, Avery Tomasco, okay, who does the local weather on the news, local news channel, and also, is very active on Facebook and stuff. And it's awesome because he has really up-to-date, personalized analysis of the weather. And so I can write this guy and ask him weather questions. And I did one today. It's not going to be a pick. It's going to be a pick later on. Where to get your data.
Nice.
It's
So
really
Avery,
nice.
hi.
It's really nice to have all of you today because the last few weeks have been slim pickings here on TWiV. Once
That's
we had
true.
just me and Alan, I think.
Yep.
Yeah, summer, everybody's off goofing around.
And I apologize for missing last week. I was on Cape Cod.
No worries. No worries.
That's
it was
too bad.
gorgeous there. It was
Terrific.
15 degrees cooler there than it was here.
Oh,
And
yeah.
that was really nice.
See any white sharks?
No, there were signs up about them, and we did see seals right off the beach.
Oh, yeah. Oh, yeah.
Just swimming along back and forth.
Were they Easter or Christmas seals?
They were wax seals.
Okay, if you enjoy this banter and these podcasts all about science,
If you don't, you're not alone.
if you don't, you're not listening, right?
That's right.
We have many science programs besides TWiV at Microbe TV. We'd love your support. We're a nonprofit entity, and we depend on your donations to keep us going. So you can go over to microbe.tv/contribute for different ways that you can help us out. And remember, if you're in the U.S., your donations are federal U.S. tax deductible. Today, so now we're almost done with July. I don't know, about halfway through. We're closing in on September, and I want to tell you about a meeting at the end of September, September 29th through October 2nd, options 12. It is an influenza virus meeting. Become fluent in respiratory virus research. I think it's more than flu, but flu will be a big one. It's called the School for Influenza. And I will be there, Kathy will be there, and Steph Langle will be there.
A lot of people go to school for influenza unintentionally.
Yeah, they do. They learn all about it. And so we will do a TWiV immune crossover. It's going to be held in Brisbane. And
Is that legal?
What's that?
Only in some states.
Yeah. Well, unlike crossover shows on your favorite TV program, this one does not mean we've jumped the shark.
I would like them to do two, but they had this idea of doing a combined one. But, with three of us, we really can only have two guests. So we'll see. I think that was the original idea. Anyway, there's a website, which we will put in the show notes in case you're interested in going. Thankfully, no obituaries to tell you about this week, because often we have too many. But we do have some news to tell you. The news is courtesy of Amy Rothfeld. And the first is, unfortunately, the first two measles cases in New York City and Washington, D.C. First one, measles cases identified at a New York City migrant shelter in Brooklyn. 2 cases. And they're trying to figure out who has been exposed. But they don't tell you much about the patients, but I'm guessing they're not vaccinated. And who contacted, where they got it from, who knows? There's also a case in Washington, D.C., an individual who visited multiple locations while contagious, and they actually have a list of the places the person went to and when and what hours, so you can figure out if you may have been exposed.
The places include a CVS pharmacy and LabCorp, so I think maybe this person was ill when they were walking around town.
Yeah, that would make sense.
Great.
Anyway, again, no information. I'm assuming this individual was not vaccinated. And they give good advice here. Get the measles vaccine, MMR vaccine, 2 doses give you lifetime protection. And they say infants younger than 12 months are too young to be vaccinated. We talked about that in a paper on TWiV not too long ago. So you see, this is the consequence of people not being vaccinated because they think it's not necessary, or they're listening to proponents who say you don't need to get vaccinated, or they say MMR causes autism, which it doesn't. And it's just going to get more and more. And then talking about vaccination, here's an article at WHO. Global childhood immunization levels stalled in 2023, leaving many without life-saving protection. 2.7 million additional children are under-vaccinated or unvaccinated compared to pre-pandemic levels, according to WHO and UNICEF.
Yeah, ironically, one of the side effects of the pandemic was that it really, really hurt immunization rates, because everything was shut down and you couldn't go do these vaccine drives. And in a lot of countries, that's the only way to get vaccinated.
And it
also ramped up the anti-vax.
That
stuff
happened as well. But in this case, with what WHO is talking about is not people who are against vaccination. It's people who can't get vaccination. In most cases.
Yeah, and it looks like a major contributor to this is areas of social unrest of one sort or another.
Of course.
Estimated 35 million children with no or partial protection against measles is a consequence of this, as well as other.
You need 95%.
You need 95% coverage to to keep measles in check. And, if this is an average of much lower than that, then you can imagine that there are pockets where it's even much lower than that.
Yeah, and these kids are probably also susceptible to other vaccine-preventable diseases. They mentioned diphtheria, tetanus, pertussis. Mumps, rubella is probably in that group because they're not getting vaccinated. But we talk about measles because that's the one you're most likely to notice first.
Somebody in one of these articles, somebody referred to measles as the canary in the coal mine.
Yes.
If it crops up, it's an indication that it's very sensitive.
And it flies very well.
It flies very well.
A paper in Nature Medicine, interesting, licensed H5N1 vaccines generate cross-neutralizing antibodies against highly pathogenic H5N1 clade 2.3.4.4b influenza virus, which is the one in cows in the U.S., for example. So the U.S. has 3 licensed H5N1 vaccines. They're stockpiled, right? And they're against different clades of the virus. There are 3 different clades. And it's not known if they would protect against the current clade. So they did some experiments to find out. They vaccinated humans with 2 of these vaccines and looked at the antibodies that they generated. And these antibodies neutralize the current clade 2.3.4.4b. So it's good, but it's not the whole story, right? It's not a challenge. It's not a clinical trial, but it suggests since antibodies are important in the correlative protection, it's a good sign because no one knew if these would cross-react.
If I ever knew that we had these things approved and stockpiled, then I forgot it. Okay,
Yeah.
That's pandemic preparedness.
It is.
Great.
Yes.
It is. And then another article which Amy wrote, Viruses Moving Without Passports. This is a weekly bulletin from the European Center for Disease Prevention and Control, so European CDC. And it has many different topics here. But the one is Oropouche infections, Oropouche human infections, multi-country. Italy has 3 confirmed cases of Oropouche virus disease in travelers returning from Cuba. Spain has 3 from Cuba as well. But the likelihood of secondary transmission of the virus among people is considered low because the vectors aren't found there.
This is quite a document.
It's very nice.
Yeah.
Communicable Disease Threats Report,
Yep.
a weekly bulletin. Holy cow.
Yeah, and there's also dengue, West Nile, dengue, Yersinia enterocolytica in raw goat cheese, cholera, respiratory virus epidemiology. Following a period of very low SARS-CoV-2 circulation, there's evidence of increased activity in primary and secondary care since May in several countries. And as Daniel said, it's increased in the U.S. as well. Influenza's here. A little bit about the Olympic and Paralympic Games coming up, mass gatherings.
Yes.
Yep.
You see, the mayor of Paris is going to go swimming to prove to everybody that the water is safe
Yes.
for the Olympics.
He's already been doing it.
Okay.
It's a she.
They've already been doing it. Yeah. Okay.
So then everyone can say the mayor is insane.
Exactly.
Oh, man.
Insane. I get it. That's good.
I looked up the current rate for West Nile in the United States. I expected more than I found. It's only in 19 states and not more than 4 cases per state. So it hasn't really started yet, to be honest.
Nobody wants to go outside the air conditioning.
Yeah, if you have it, if you live in Texas. Hey, Rich, if you live in Texas, you don't have air conditioning. Have they recently solved that?
Oh, you mean in Houston?
No, in anywhere else. There was like 150,000 people without electricity.
Yeah, that was in Houston after
Mostly
the,
in Houston, okay.
Mostly in Houston after the storm.
I presume
that was supposed to be fixed by now.
Yeah.
I don't know. Fortunately, well, for better or worse, that storm missed us. We were kind of hoping for an edge of it to give us some rain.
Yeah.
Okay,
That's
but
right, that's right.
it went east and clobbered some other poor
Yeah,
unfortunates.
but
Oh
they
yeah.
got rain in Florida, in Vermont.
Oh yeah, they got so much rain here on campus one day, and a subterranean part of a parking garage
Yes.
flooded and
Oh,
totaled 30
no.
cars.
Oh, no,
Mm.
no. Oh, my
The pump
gosh.
failed, and yeah, it was a mess.
Oh,
They
Oh, my
couldn't get
gosh.
people to move their cars in time. Yeah.
Yow.
All right, on to our literature for today. We have two interesting papers, which will both bring up conundra of nomenclature, as you will hear. And the first is published in the PNAS, recombinant OC43 SARS-CoV-2 spike replacement virus and improved BSL-2 proxy virus for SARS-CoV-2 neutralization assays.
I'd like to give the short
Oh,
summary.
yes, please.
Very short summary, which is they have taken one of the common cold coronaviruses called OC43, and they have put in the SARS-CoV-2 spike. And what that does is attenuate the virus, give them possible vaccine candidates, and allow them to work with this virus at BSL-2, a reduced biosafety level, which means a lot more experiments can be done.
yes.
So
And
OC
I'll
stands
do
for over the counter.
Old cow.
right. It's Old Cow 43.
Old cow. Oh, I see.
So the authors, Je Hu is the first author, and Julie Fox is the senior author, and they are at NIAID, the NCI, and the FDA. This is an all Maryland team.
Uh-huh.
Actually, Jonathan Udell is the second last author, and he's
Oh.
one of the people who wrote the paper.
Yep.
So
Yes, okay.
All right, so as you may know, if you don't, you will learn, SARS-CoV-2 must be worked at under BSL-3 conditions, Biosafety Lab 3, which is, not everyone has a BSL-3 lab. The labs that Rich and Kathy and I and Dixon had are BSL-2 laboratories. BSL-3 is another level up where you have to wear suits with filters, pressurized filters, and so forth. So it's kind of
This is one step below the BSL-4, where you're in the full spacesuit with the airlock and everything.
But it's limited what people can do. And so one of the things that you want to do during, well, for any virus, you want to do neutralization assays where you take whatever virus is circulating, and then you take serum from people or animals and ask, do the antibodies block infection? And you need infectious virus for that. You can't do it with bioinformatics. Sorry, you can't do it by sequencing. You have to do infectivity experiments. And so what people have done to avoid using SARS-CoV-2 is to use pseudotyped viruses. And we've talked about these. You take it, we'll discuss what pseudotype means actually, but instead of using SARS-CoV-2, the main target for the antibodies is the spike glycoprotein. So you take the gene encoding the spike glycoprotein and you put it in another virus, you replace the other virus's spike glycoprotein.
And this is generally done with a completely different
right,
virus.
Like vesicular stomatitis virus or a lentivirus like HIV, where in case of HIV, it's been gutted, so it doesn't pose any threat, and VSV is a BSL-2.
Got it. As if it had a gut.
Gutted, yes.
Yes.
And so what they have done here is that let's take OC43, a human endemic common cold coronavirus, which is benign in most people. Not everyone. It's benign in most people. And can we substitute the spike glycoprotein with that of SARS-CoV-2? And it'll be a BSL-2 pathogen and use that for neutralization assays and maybe some other things as well, actually.
This is a really clever approach because it's not just pseudotyping. It's you're transferring this gene to a very closely related virus. So it's going to be presented in a context that's much more similar to the way it is normally presented. And they talk a bit about this, how when you put it on a pseudotype virus like a VSV with spike, there's the spike dangling out there and it's not really in its normal context. And the responses you get are going to be a little bit different. Whereas this, putting it into another coronavirus, should give us a much better context for presenting this antigen.
So The word pseudotype or pseudovirus. Let's talk a little bit about that. I don't know the
It's
origin.
not a pseudovirus. It's a real virus.
Yeah, no, it's a real virus. I don't know, but it doesn't exist in nature, right?
Right.
So I guess that's where the idea pseudovirus comes. Kathy, you have any thoughts about that?
I don't. It's actually, those terms are both used in the second paper that we're going to talk about. They're in the graphical abstract. There's the pseudotype variant library, and then there's the pseudovirus.
Yeah, the only thing I would warn people is don't think it's not an infectious virus because it's called the pseudovirus, right?
Right.
The virus gets very insulted about that.
So I brought up in the show notes to everybody, why is this what we're talking about in this paper for the OC43 SARS-CoV-2? Why is it not just a chimera?
Right. Right.
That to me is just a choice of words, right? They call it a recombinant versus a chimera, right?
Well, they call it a replacement virus and a proxy virus, and
Yes.
it seems very odd to me that they don't just call it a chimeric virus.
I hadn't heard proxy virus before.
Yeah.
If we had
Anyway.
Jonathan Udell on, he could take an hour to explain why they did it, but he's not here, so maybe he will write in if he's listening.
We're going to get mail.
I hope so.
So the first step is they take OC43, which is an RNA virus, right? It's a coronavirus. They take the RNA, convert it to a DNA copy. They put a fluorescent reporter virus in the genome so they can track it easier. And then they use that to recover infectious virus. So that's the starting point. You need a DNA copy because then you can cut out the spike gene the spike gene of OC43 and put in the spike gene of SARS-CoV-2. And so they make, this is very interesting, they make the infectious DNA of OC43 at BSL2 because that's what the virus is rated at. And then they move to BSL3 to make the recombinant because they don't know if it's going to be safe, right? Which is good. That's the way you should do it.
So you should call it Lego virus.
Yes.
But just
right. So you treat it as based on its most dangerous ingredient.
And so I want folks to understand that scientists do this. They consider the potential threats. We're going to see this in the next paper as well.
Mm-hmm.
They don't just do stuff that they want to do without any consideration of safety, which is which impression you might get from listening to the news. They're very considerate about safety. So this is an example of that.
Well, because remember, the first people who would get hurt by this are the scientists working on it. So yeah, we tend to be kind of careful.
We're not cowboys or cowgirls. We're just being careful. So they go into the BSL3 and they construct the recombinant. I would call it a recombinant virus.
That's
right?
good.
Yeah.
Because that's what we used to do. We used to take the DNA, you cut out the spike gene, you put in the SARS-CoV-2 spike gene, and then you can recover virus from that DNA in cells in culture. And then you sequence it, you make sure it's what you think it is. They use monoclonal antibodies to confirm that the OC43 spike is gone. It's replaced by the SARS-CoV-2 spike. Interesting, they do an experiment. They pass the virus 7 times in Vero cells, and it undergoes changes. 12 amino acid deletions that remove the furin cleavage site, interestingly, the polybasic cleavage site at the S1 and S2 border. And there are other changes as well that affect different proteins, but it doesn't affect the replication in the cells.
And these changes, as I understood it, are things that they have seen, that have been seen before in SARS-CoV-2 that's been passage in culture. So there are pressures on the virus in culture that select for these sorts of things.
And these results suggest that this recombinant virus is behaving, if you will, the same way that SARS-CoV-2 would in these conditions.
They should.
Vincent, just to get at your idea of you would call it a recombinant virus, I think that's why their constructs have a little r at the beginning of them. I think that's for recombinant.
Yeah.
That's my guess.
Correct. Yes, ROC43-CoV2S. Correct. So they show that the tropism of the virus has changed in terms of receptor utilization. This virus binds to, so the parental OC43 binds all cell types regardless of human ACE2 expression. and this one requires ACE2, which you would expect from the SARS-CoV-2 spike. They find that it replicates a little more slowly than OC43, which is not surprising because this is a human-made virus and doesn't have the fitness that would come from evolution in its natural host.
They messed with it. Yeah.
They messed with it.
So you might want to call it a designer virus or a custom virus. Perhaps
This
a
is what we
custom
need
virus.
is even more vocabulary.
More
to
names.
use.
Yes, right.
Yeah, but you'd never be confused about it, though.
Well, this
We're
is
confused about
and
this one.
Let me ask the group here: is this a gain-of-function virus?
Well, yeah,
Sure.
yeah.
Finds a new
because
receptor, right?
you've
Do we have to
you've
answer that
changed
question?
you've changed the receptor specificity
Yep.
at least of the background virus
Yep.
OC43.
That's right.
It is very much a gain-of-function experiment.
Mhm.
So just keep that in mind. It's a good thing. This has very interesting properties, this virus, and I think it will be very,
That's right.
very useful.
And if they patent it, it'll be a gain of income virus.
Right.
I'm going to drop out of this broadcast, I think.
They find that antibodies against S will block infection with this virus, right? Which is what you would expect. And they also make versions with the spike from Delta and Omicron, and those are going to be useful later on. Again, they mention that it replicates to a slower rate and to lower titers than SARS-CoV-2 in Vero cells. And temperature difference is interesting as well. OC43 with the spike grows better at 35, which is the upper tract temperature, than 37, lower tract, whereas SARS-CoV-2 grows better at 37 degrees. So different optimal, which is presumably caused by everything else in OC43 besides the spike, right?
Right.
So they conclude this is a highly attenuated virus, and most likely we could use it at BSL-2, but they then do some pathogenesis studies in mice. They infect ACE2 transgenic mice, and they use this virus as well as SARS-CoV-2. The mice infected with SARS-CoV-2 get sick, and some of them have to be euthanized because they get so sick, whereas the mice infected with OC43 CoV-2 were indistinguishable from mock inoculated mice. They didn't lose weight, had no mortality. They looked at infectious virus in different tissues. SARS-CoV-2 reproduces to high titers. For the recombinant, they found no infectious virus or viral RNA in any organ for 7 out of 8 mice, with minimal replication in 1 mouse, if anything. So it's highly attenuated in mice. They do staining of lungs and see very little pathology caused by infection with this recombinant compared to SARS-CoV-2.
They have a nice little rap about comparing RNA to virus.
They do.
Yeah.
I had highlighted that. I wanted to. Is that later in the discussion?
It's probably later. I think there's actually
Yeah, I wanted to focus on that. Yeah.
They do it here, but they may not comment till later.
So in a mouse model, the virus is also highly attenuated. What about neutralization assays? That's why one of the main reasons they designed this. So they mix either SARS-CoV-2 or OC43-CoV-2 with antibodies and measure neutralization titers. And this is really interesting. It's really hard to neutralize. It's tenfold harder to neutralize the SARS-CoV-2 compared to OC. Actually, they use VSV as a comparator because that's what most people use. VSV COVID with the spike of SARS-CoV-2 compared to their virus. And their virus is much more sensitive. And that makes a lot of sense to me, right? Because OC42 is a coronavirus, whereas VSV is a different virus family. As Alan said earlier, it's different architecture of the spike on the surface and so forth. The reason and they say this is a major improvement over VSV CoV-2 pseudotypes due to recapitulating the spike organization on the virus surface. You know, I've asked people for a long time, for a few years during the pandemic, are you sure that this VSV spike is okay? And everyone says, yeah, of course, it's the people who are using it. Yeah, it's totally identical to regular virus, but it's not. It's not clearly. So I hope people use this. This seems to be.
Yeah.
Mhm.
And interestingly, if I recall their discussion correctly, it's the VSV pseudotype actually has a much higher density
Yes, that's
the
right.
SARS-CoV-2 spike protein on its surface than does the real virus. You would kind of, just naively, you might think that would be a better thing, but maybe not. Maybe it's got
Yeah,
too
I mean,
much.
if it's all crowded together,
Yeah.
you may not have as much
Hard to
antibody
neutralize
access
it.
to
But
it.
it's also a bullet, right, compared to a sphere.
Yeah.
So that may
Yes.
make
Sure.
a difference as well.
It's like quite different.
And then if you use the lentivirus, it could be different as
Different
well.
still.
So, and then the next experiment, they say maybe this would be a good attenuated vaccine because it doesn't cause any disease in mice as far as we can tell. So they immunize mice, and then 29 days later, they challenge them. If you do this with SARS-CoV-2, with naive mice, they get very sick, and you have to kill many of them. Mice get a single dose of this recombinant virus, intranasal immunization, by the way, 100% survival, no weight loss. It's really looks good. They even do an experiment in macaques where they immunize mice intranasally with this virus, and
Immunize the macaques.
then they look at the antibody response, and they find a single mucosal dose gives you strong spike-specific IgA and IgG, which neutralize the virus. But what's interesting here, this is where the RNA and virus come in. So
Yeah.
they measure infectious virus and by plaque assay and also by RT-PCR and nasal swabs. And they say infectious virus was not detectable after day 1, but they could detect RNA. And they write the discrepancy between viral RNA and infectious virus is observed in many viral infections. And they give two citations and may be indicative of abortive replication and or the generation of defective virions. So, we've said so many times that just because you find RNA by PCR doesn't mean that it's infectious virus. And this emphasizes that. And it's known.
This was a problem early in the pandemic.
It
Yes.
was, yeah.
When the PCR test was the only thing around. And, basically, this is not faulting anybody. It's just that, it's what the information and the technology that we had available. But they didn't want you going around if you were PCR positive. And it can take forever to clear the RNA.
Some people were, depending on their country's own policies, they were quarantined for months because they were still PCR positive.
I think, part of the problem is we didn't have a better assay at the time.
Yeah.
But also, people assumed that you were infectious. It was the default assumption without looking at the literature and seeing that for other viruses, you can have RNA without infectivity for many, many days after infection. So, but even with antigen tests, that's the same situation. You can have persistent antigen and no infectious virus. So, needing a BSL-3 to do infectivity is a real problem, right? So that's what they find. This is a surrogate, a replacement for VSV-based pseudotypes. It seems to be more sensitive, and perhaps it's a attenuated vaccine candidate.
And they have this has now been approved for BSL-2.
And it's BSL-2. That's right.
So
And that'll be real handy.
based on the analysis that they had done, it's now been cleared to use at BSL-2, which is just your ordinary lab bench.
At their institution.
At their institution, yes.
Well, yeah, but the committees are committees that look at this stuff from other institutions as well, including basically, the NSAB dudes.
Right.
Yes.
As I understood it. So in terms of the use in particular, terms of use as a live attenuated vaccine, the question, of course, that runs through my mind through all of this is, what about the T cells?
Right.
Okay. And I wonder if there's any cross-reactive epitopes between the cold virus and SARS-CoV-2, and whether T cells, what role T cells play in all of these phenomena.
Didn't Crotty early in the pandemic show that some people had cross-reactive T cells, and the presumption was they were from common cold coronavirus?
Yes, I believe that's correct.
Yeah.
I believe that's correct. There's some, at least minimal cross-reactivity.
But now you have a reagent that you could use in experimental animal models to look at that further. Yeah. So I think it's quite a nice paper, a lot of basic virology and nice outcomes. So thank you very much for that.
I'll elaborate just. Go ahead, Kathy.
Well, I was just going to point out that they also look specifically at IgA and IgM antibodies, and they find that there's good generation of those with this as well. So that they point out that could make it a better vaccine from the standpoint of getting at the virus when it first is at the sites where it's first entering.
Mucosal immunity. Yeah.
Right.
So, actually, it turns out I got two more things.
Yep.
Okay. The first is that they puzzle over the fact that they get protection and a pretty good immune response, even though they can detect little or no replicating virus. And they actually make a comparison to the Mpox vaccine, MVA, which doesn't replicate to any significant extent at all. And to me, this means that probably with maybe not even a complete round of replication, but an infection of cells and gene expression without generating virus in a dose of a vaccine is sufficient to give you a protective immunity, which speaks to me to the robustness of the immune response. If you like, you don't need a lot of antigen. Okay, it's not like you have to have a lot of virus spread. I find that interesting. The other thing I thought about when I was thinking about the T cell response, in my mind, I was making a comparison with the dengue vaccines because this is a little different, but the dengue vax was the dengue surface glycoprotein stuck into the yellow fever virus vaccine backbone, related virus. Okay, not probably nearly as closely related as these guys, though I can't actually make the comparison. That didn't work all that well. Okay, what did work well was having the attenuation in a dengue backbone, which tells you that there's other things going on than just the immunodominant epitopes. It speaks to the T cell response. So I was thinking about that as I was wondering about the T cell response in this recombinant virus.
So I'm wondering how this compares to OC43, right? We don't have those experiments. I don't think there's a model for OC43. So why is this virus attenuated? Is it actually more attenuated than OC43? Because remember, OC43 does make some people very sick. So is it that these additional mutations in the genome attenuated? Is it having the other spike incompatibility with some of the other proteins? It would be interesting to understand that. And I think before you could put it in people, you'd need to know more about that as well.
Yes.
And by the way, I have a coronavirus here that was sent to me by Mark Martin. This is a glass, a lady on Etsy made this, a glass blower. And
Very nice.
It's beautiful. It doesn't stay in focus, unfortunately, but it's got a little red in it. Isn't it lovely? It's solid glass with little
Very
spikes.
pretty.
So those spikes, that's what we're talking about.
It's gorgeous.
It's gorgeous. Yeah. Thank you, Mark. Okay. Our other paper is published in Immunity. Deep mutational scanning reveals functional constraints and antibody escape potential of Lassa virus glycoprotein complex. So Dixon, I was thinking of you when I selected this paper. Got Lassa virus. You could tell us some Lassa virus stories, perhaps. This is two first authors, Caleb Carr and Catherine Crawford. This is from the laboratory of Jesse Bloom. And other authors include Murphy, Galloway, Haddix, Matson, Christian Anderson, Neil King, and Jesse Bloom. So it's from the Fred Hutch in Seattle and the University of Washington, the Scripps in La Jolla.
And Caleb Carr is not the novelist
No.
who died a few years ago.
And Christian Anderson is not the person who writes children's books.
Neither
right.
is he
Yes.
the
Couple
Caleb
of very
that's at
difficult
the
to
beginning of our show notes where we say, remember,
Not that
Caleb is
Caleb.
listening and cherished too.
Well, actually, do we know this is not that Caleb?
Yeah.
We don't.
Because that
You
line
know.
has been in our show notes for a long
long
time,
time.
and I often wonder, what is Caleb up to these days?
He was a
So-
He was a sixth grader, I think, or something like
He
that, or
was.
maybe even
Yeah.
younger when that first came up.
And that was what, like 10
More.
plus years
Easily
ago?
10 years ago.
My gosh.
Yeah.
So this could be, I don't know. Caleb, if you're
I
listening.
must say that I was encouraged when I read this article when I went to the first reference, and it was John Frame's reference.
Mm,
That means that they're honest people and they're giving credit where it's due.
John Frame. Let's see, reference one. Let's take a look.
Number
Frame,
one.
Lassa fever, a new virus disease of man. I wouldn't say man anymore, right?
Yeah, well.
From West Africa. One, clinical description and pathological findings. You know, it could be a really interesting paper published in 1970.
Yeah.
We could review
You know,
it if you want.
fundamental stuff. No sequencing, no deep
No.
mutational scanning, which
It
is
was all epidemiologic.
Observations, pathology.
Yeah.
That could be fun to do.
Yeah.
Yeah, yeah,
We should do that. Anyway, so
Tom
Yeah,
Monas-
as long as you're on that for
Yep.
original references, they also reference the discovery of the animal host,
That's right.
which
have
was
Tom
a
Monas reference there
principal,
also.
and that was Tom Monas. And they
That's
who was
he's
on
been on the
TWiV?
show.
He talked about that, right?
Yep.
Yeah.
Right.
And that animal host is Mestomus natalensis, which
That's
is
the
a mouse.
multi-mammate rat. That's right.
Yeah. The first four references are all from 1970, and number 3 includes Jordi Casals.
And it was right, exactly right.
Nice.
Who almost died from it.
Mm-hmm.
Yep.
Oh, it's
And
a
by the
wonderful
way,
story.
this paper and the other one we are talking about today are both open access.
So Lassa fever caused by Lassa virus, thousands of deaths every year across West Africa. Nearly every case is a spillover from the Mastomus natalensis, although there's a little bit of human to human transmission, but mostly, it's like West Nile. Every human infection is a spillover. And so a lot of deaths, no vaccines, no therapeutics. And one of the problems is there's a lot of diversity of Lassa virus with 7 lineages over distinct geographic regions. So how that impacts therapies like vaccines, antivirals, monoclonals, and so forth, we don't know. This paper,
Well, the fact is there are no antivirals and there are no vaccines.
That's right.
Right.
so
So
if
Now,
you
we,
had
that's how
some,
we
different
do
is
know there is one encouraging note here,
Mm.
which is people have found antibodies in people who've survived Lassa virus,
right?
and those neutralize Lassa virus. So that suggests we might be able to develop a vaccine.
So I'm telling you now that was John Frame also, who was sent to Africa virtually every year he was alive to collect immune serum from people who had recovered from Lassa fever. And that was sent back to all the laboratories that were working on Lassa fever at the time to make sure that if there was a laboratory infection, they could save their lives. And Jordi Casals was the poster child for that.
He was treated at Columbia Presbyterian in New York, right?
The nurse Pinio was the woman who recovered naturally.
She recovered, and then they gave him her serum.
That is correct.
And you
That is
wrote the
correct.
book about this, right, Dixon?
I did not.
No, you wrote the West Nile story. Sorry.
That's right. I wrote
But
a
we
different
did.
book.
Actually, the first episode of TWiV was
West Nile.
was
virus.
West Nile.
West Nile.
Then we
That's
did
right.
a
Yeah.
Lassa episode. I'm getting it all confused.
That's right.
You can't be president, Nixon.
Anyway, I'd like to be sent somewhere every year to do work. That would be fun.
Yeah, he enjoyed the hell
Oh,
out of it too.
that's
I mean,
cool.
he came back all smiles.
So the glycoprotein complex, just a spike sort of protein that we've been talking about for SARS-CoV-2. That's the main target of neutralizing antibodies. We know as...
And we should probably mention Lassa virus is a single-strand RNA virus with an envelope, right?
It is. May I have the envelope?
Yes. So
it's a complicated dude, actually. It's arenavirus.
It's an arena.
Two segments, each ambisense.
Yeah.
Okay, so both senses are used. It's a complicated dude.
It's a little
so
minus,
hard
a
to
little
teach
plus.
that to
I
students, the
know.
whole ambisense thing. Oh.
Yeah.
So we know
So this-
antibodies that target the spike are made and can neutralize infectivity. Vaccines are being developed. I've spoken with a number of people over the years on TWiV about those, but we don't have anything yet. And the purpose of this paper is to try and ask, in this spike protein, the GPC, they look at every amino acid and say, which of them can vary in terms of attaching to the cell receptor, right? So the spike attaches to the cell receptor, but also how many variants could you get and make resistance to the antibodies, right?
Yeah, we often talk about how viruses are very constrained systems and they can, you can mutate, for example, the SARS-CoV-2 spike, but it can't mutate too much, or rather you can mutate the gene for it. Sorry, Vincent. You can't change it too much because then the virus can't bind its receptor. So what they're doing in this paper is drawing a map. What can change and what can't in this GPC.
The remarkable thing from my perspective, and I mean, I don't know anything about this except to look at that graphic for a map function constraint throughout GPC, and it's a beautifully graphic illustration. You don't have to know anything about viruses to find out what the best part of that spike was for developing antibodies against it. That's a remarkable insight into how to present data. I like that very, very much.
Yeah, the punchline of this paper is very easy to grasp. So the way they got there is a little more complicated because
well, that's right.
How do you figure this out?
So they use pseudotyped viruses.
Yes.
In this case, they're using lentiviruses, right? So this is an HIV-based vector where they put in the gene for the GPC of Lassa virus, and these viruses are quite simple. They are just the gene for the Lassa GPC, and there's a little bit of HIV on each side, but none of the HIV proteins, right? Just
it's been gutted.
Long terminal repeats and some other elements, promoters and so forth. And so this is not going to reproduce multiple times. It's going to be one, you infect cells and the glycoprotein will be made and that's the end of it. So it's a BSL-2 experiment, whereas Lassa is what? BSL-4, right?
Yeah, I think so.
I think they say that.
Yes, it is 4.
Yeah.
So this is making it easier to do these experiments. And so deep mutational scanning basically means every amino acid of how many amino acids in the spike? Anybody remember?
A lot.
9,820 amino acids. They mutate every one to every possible amino acid.
No, wait a minute. I did this calculation.
Possibilities.
Yeah. There's like, it looks like just short of 500 amino acids.
Right.
That's better.
And with 20 possibilities in each position, you're talking about a bit over 90,000 possible amino acids total.
9,000.
With 20 possibilities of each of 500 positions. Okay.
9,820 possible amino acid changes. They call them mutations, but they're changes. They're changes, Jesse. I'm not going to argue with Jesse. You don't get anywhere when you do that.
You're just going to correct that here. Yeah.
So you have these lentiviruses with the GPC gene in them, and then you subject them to PCR-based mutagenesis, basically, to make all the different possibilities at each position. And in addition, you put a barcode in so that you could identify each spike by sequencing it. A barcode, it's a sequence that's unique to each change. And then you generate stocks of these viruses. And here, now before we talk about the results, here they make this paragraph of discussion, which is quite interesting. They say, okay, the natural host of Lassa virus is this Mastomus natalentus rodent. And so the glycoprotein is optimized for binding to the rodent receptor. It may not be optimized for infecting humans. And we're going to make changes that could optimize GPC to bind humans. And we're worried about that. So if in fact we do find such mutations, we're not going to publish them. We're going to only study the rodent mutations, because someone might say, here's a mutation that encourages it to bind human and use that for nefarious purposes and so forth.
Right. So
Right,
they would
right.
like to study this in human cells, but first they check to see if they're going to end up improving binding to human cells, because that would be in a direction they don't want to go.
Right.
Right. And so they talk about this very specifically as that it could generate potentially hazardous information. And they give
Yes.
references to 2 papers which discuss the idea of how now there's more danger from biological information than from biological samples, and how that ought to be treated. And so then throughout this, the authors of this paper go through and describe how they checked this out very carefully and determined, as we're going to find out, that there wasn't any difference between the results for human and the rodent. And so they could go ahead and go forward with studying it as in the human form.
So they
Yeah.
first address that question: is are we going to improve the use of human receptor, which is alpha-dystroglan, by the way, is the receptor, but it's different protein in human and rodent cells. So they take cells that either produce human alpha-dystroglan or rodent alpha-dystroglan, and then they infect them with their pseudotype virus, and then they find they actually have the whole library constructed already, and they infect both of these cells. And when you do that, you get enrichment for the viruses that are viable, right? So many of the amino acid changes are going to destroy viral infectivity. And so you can, as we'll see in a moment, you can calculate the effect on fitness. But they see no difference in the human versus the rodent receptor. So they're confident to go ahead and use human receptor for their experiments.
Right. And because this initial experiment is being done with a pseudotyped virus, you don't have to worry that you're actually making a more human-suited virus in the process of this. All you're doing is determining whether the receptor on this gutted lentivirus can bind better to one or the other. And it can.
So the way the experiment is done, they have this library of pseudotype lentiviruses with GPCs. Each have mutations in the gene for GPC, and they infect cells, and then they sequence what is produced, what is in those cells, to get an idea of what changes work and what don't. And they're barcoded, so they know exactly what amino acid change is making it or not making it, right? And so they do a score, they quantify, and they use cell entry as a metric, right? So if the if the pseudotype can get into cells, then that means the GPC is working to bind the receptor and get into cells. And so their cell entry score, so they can sequence the barcodes and they compare their library to an unchanged glycoprotein, and they say like a score of 0 would be parental entry, and negative scores would be impaired entry. So that's how they can do it. And so they basically say, where are the changes in the GPC gene that allow entry into cells? And many are deleterious to entry. Some of them, though, are tolerated. And in particular, the GPC has different domains. There's a stalk, and then there's a head of the glycoprotein. And they say the stalk is less mutationally tolerant than the head. It's shades of influenza virus, right? Where the HA globular domain is more mutationally tolerant than the stalk. Then there's a pocket at the tip of the GPC, that binds the receptor. And they say the core binding pocket is mutationally intolerant, which makes sense because it has to bind the receptor. So if you change amino acids, it won't be able to bind. You're not going to get entry in your assay.
I just had a revelation. When they were saying this in the text, I kept going and looking at the figure. I was looking at part A of figure 2 and trying to look at the shades of orange or brownish versus blue. And if you look at B, it's very clear that the
Yes.
stalk is not tolerant and that this little pocket in the very top
Yeah,
of the
yeah.
trimer is.
I wonder if in those cases where you have a stalk mutation, if the protein actually even assembles correctly in the membrane.
That's a good question. Yeah. I bet that's a fraction of it. Yeah.
I looked at that same figure. If you go to B in figure, let's see, where is this? B, it looks exactly like a tungsten light bulb.
Yes.
Yes. Yeah.
Not an LED, Dixon.
Not an LED. Well, they're making LEDs to look like
They
tungsten
are.
light bulbs now.
Yes.
Yeah. By the way, speaking of LEDs, so I have this big light here that's illuminating me. It's really,
Yes.
it's about 2 feet in diameter. It has a white filter on the front, and there's an ant crawling up and down it. I've just been watching this guy go back
Casting
and forth.
shadows on
He's
your
walking
face.
on the backside of the white diffuser, and I'm like, I really feel bad for this ant because it's
Well-
not going to get anywhere in that light, right?
Well, you need to go rescue him after the show.
Yeah.
That's right. That's right.
Well, that involves taking the light apart and everything. No, but when I find spiders in here, I'm in the basement. I pick them up and bring them outside.
Sure.
Good heavens.
I mean, they have a right to live, don't
They've
they?
got DNA, man.
Tell them there's an ant in your lamp. It'll get it.
I like spiders. They eat mosquitoes.
You don't
That's
like
true.
I
mosquitoes.
also think they keep down the roaches, I think.
The spiders do?
I think
do.
so. That's
Maybe.
what-
Oh, yeah, they might.
Many people
absolutely.
don't like spiders. They think they're dangerous, but most spiders are not, right? They're just a couple.
Okay, one of the coolest lab things that we did when we went to the Okefenokee Swamp was figure out, or somebody knew this, if you hold a flashlight right beside your eyes and
Oh,
point
yes.
it,
Yes.
you can find spiders because
Correct.
they will light up green. I guess it's their eyes.
And they have
Anyway.
orange backs also that also
Yeah,
light up.
but
Wow.
you see this bright, almost fluorescent green, and so you
Cool.
can go in your garage with a flashlight at the
That's right.
level of
That's
your
right.
eyes
That's right.
and
That's
look
right.
around and see all the spiders.
I'm going to have to try that in my basement.
It's really cool. Yeah,
Huh.
Neat.
yeah.
But we digress.
Okay,
Yeah,
back
we
to
did.
the paper. So
By the
we
way,
digress.
a number of arthropods also fluoresce in UV light.
Yeah, I'm
So
not
if you-
exactly sure it's fluorescence, but it's definitely
Right.
something.
No,
it's
this
that
would be
bright
this would be a
green,
different
yeah.
a different
Rich.
thing. You're
Yeah.
talking about regular flashlight.
Yeah.
But if you have a UV flashlight, which is pretty commonly available for a variety of things like finding pet urine stains, that kind of thing, you can, if you look around with that, it'll highlight the scorpions certainly, and then there are some other arthropods that
That's right.
also glow.
So, Rich, this digression was known as a loss of function.
Yeah, but now I got a bunch of experiments to do in my house.
Yeah, it's
Yes.
really good practical information.
Yeah.
Yeah,
There's
yeah.
lots more information about spiders in the web.
Yeah,
Here, here.
Ooh.
yeah, Rich. But
Yes.
do the flashlight one with your grandchildren.
Okay.
Yeah, that's right.
Yeah.
That's right. That's right.
So what do they use this library for now? We know that there's a library. You can see which amino acids can be changed. They measure, the first thing they do is antibody neutralization, right? Antibodies bind to the spike to block infectivity. So they ask, can we identify residues that are involved? So what they do is they take their lentivirus pseudotype library, like lots and lots of lentiviruses with different changes in the GPC gene, and they mix it with antibodies before they infect the target cells, right? So if a spike can be neutralized, it won't get into the cell. But if an amino acid change has made it resistant to neutralization, it will get into the cell, and they can measure that with their barcodes. So first, they have an antibody that's well characterized, known to bind to the very tip of the GPC, and they can find escape changes in their library that block neutralization with that antibody, and they're concentrated at a small number of sites in GPC in a known epitope, which is the binding site for the antibodies. And interestingly, So some of these changes allow entry in their previous assay, the entry assay, but not all of them. So you may have an amino acid that blocks antibody binding, but if it doesn't allow virus to get into cells anyway, it's useless, right? Well, it's not useless, but that virus is never
So
going
this
anywhere.
again is mapping the constraints. The
Yeah.
virus could, if it had this change, this amino acid change, it could evade the antibody, but then it couldn't get into the cell.
But for some, there's clearly a change that will allow entry and allow resistance. So they say the potential for escape from this antibody with little cost to GPC function, right? So if the GPC doesn't work, the amino acid change, even if it gives you resistance to neutralization, is never going to propagate in the virus population, which, I mean, it's not good for us. It's good for the evolution of the virus, right? So then they did this with 6 neutralizing monoclonals that have been isolated from people. And they say all of them escape neutralization by the same changes that have little impact on cell entry as determined by their previous assay. So they conclude that the glycoprotein has substantial evolutionary capacity to escape each of these antibodies.
So I'm in my mind, I'm generalizing here, okay? Because some viruses, and I'm thinking measles, show very little antigenic variation.
Correct.
And I attribute that to, and I'm sort of checking myself, make sure I got this right. I attribute that to the notion that if you change the antigenicity, okay, it compromises the function. So there's a fitness cost for changes, and that constrains the virus to a certain narrow window of, serotypes, actually.
Yeah.
And viruses like this can do a pretty significant amount of change without affecting the function of the protein, so they can drift all over the place. Really, you would think, why doesn't measles get its act together? Okay.
Well, it's very,
why doesn't it have a more flexible
but it's very
receptor?
good.
Yeah.
It's fine. It does well without having.
It does well. Well,
Yes.
yeah,
Right.
it compensates
Do you remember
by being
the paper
incredibly
we
infectious.
did a paper where we did this a similar experiment with the spike of measles virus, right? And they looked at the parts that could change. And Alan, remember, it's like they used to look at the planes coming back from and
Yes.
see where the bullet holes were, and they make that strong, right?
So
Right.
that's it. I typed that into the show notes here. I didn't want to interrupt earlier,
Oh.
but both when I read it and when you talked about it, I was reminded of that World War II mapping of
Yeah,
the
you
vulnerable
reinforce the
parts.
areas where you
Yeah.
don't see bullet holes in the returning aircraft.
Maybe
Right.
that's the
Because those were the holes in the ones that didn't come back.
Right.
maybe that's the episode image.
I think it was. Yes.
You
It's
reinforce
hard to
the areas that did not have bullet holes, right?
Yeah.
Yes.
Right.
Yeah, because the bullet holes are redundant or they're not a problem. Yeah.
Those are planes that survived.
Interesting.
Yeah.
Okay, so they do this analysis. They say most of the escape mutations are at site that fall in the footprints of the antibodies that were previously determined by structural studies, right? But they say also sometimes amino acid changes in the footprint didn't affect neutralization at all, yet the viruses could still get into cells. So that's very interesting that some changes will not affect neutralization. And most of the changes are in the footprint, but they say there's some sites where we could change an amino acid and it would affect antibody binding, but it's distal from the epitope. It's distal from the antibody binding site. And there were 2 kinds. One, some of those amino acids actually affect the attachment of sugars, which can impact whether an antibody will bind or not. But other sites were below the surface of the glycoprotein. So most of these epitopes are on the surface and the antibodies just contact them, but some were below, so they must be deforming the surface so the antibody can't bind.
The thing that strikes me most about all of this talk that we're engaged with is that nature creates a scaffolding, in this case, a light bulb, like projection from an object. And only a very, very, very small portion of it is involved in the actual action. The rest of it is all scaffolding. It's just like, I think I'll build a light bulb that has a receptor on the top. or an entry molecule. It's that way with a lot of other things too. It's not just viruses. It's the way enzymes work. It's the way proteins on your surface work. It's remarkable how nature needs to construct a building that only has, like, say, one door or one window. I'm fascinated by that.
Yeah. Now, here's an interesting observation. They have this, there's an antibody cocktail that's being developed to treat infections. It's called Aravirumab 3. 3 antibodies, 3 monoclonals are in it. So they looked at what changes would lead to escape to these 3 antibodies. 2 of the antibodies can be escaped from by the same amino acid changes, whereas the third, you need different amino acid changes, right? And so they say that's what makes this good, is that third amino acid, that third antibody needs a different change from the first 2, and that makes it less likely to happen, right? And the worst scenario would be if you had 3 antibodies and they all are changed, you get resistance to all 3 with the same amino acid change, then you're screwed. So the point here is that you have to figure this out ahead of time before you use it in people. And this is a validation of this approach. I think it's cool.
Yeah.
They also find some of the amino acid changes that they discover, they look in public databases and they find them in Lassa virus isolates. And so these changes are already out there, which says, if you had a patient, maybe you'd want to know what sequences were in that glycoprotein there before you treat the patient, if you had more than one option anyway, because it's like with HIV. When you have a HIV diagnosis, you sequence the isolate, make sure you're going to give the patient the triple combination of antiretrovirals that will be sensitive to, because there's a lot of resistance out there. So this is the same idea. I think that's pretty neat. So they say in the end, we can predict the escape of natural sequences simply by summing the effects of their constituent changes as measured in the deep mutational scanning. So they made this interactive phylogenetic tree. They use NextStrain, which you may remember from previous TWiVs. So you can go in and search for an amino acid and see what the predicted escape would be. So maybe it's useful for designing vaccines and monoclonal antibodies.
Yeah.
Right. Identifying sites in the protein that are likely to be tolerant or intolerant of future change. Right. So if you have a site that can't change, maybe that's where you want to direct an antibody, or make a vaccine that directs antibodies against that. Right.
Or you at least want a vaccine that generates enough different antibodies consistently that the virus can't mutate or can't change around them.
So they also say if you want to design vaccine immunogens, you can look at and we talked to Jason McClellan about this, Rich, some time ago in Austin, how you can make designer vaccines where you introduce amino acid changes in the immunogen that have particular effects. And they said, here we can tell you which changes are going to mess up the folding, because it makes a protein that doesn't get into cells. So it is a really good resource of information that people that want to do that. Oh, and the other thing I didn't mention, these 6 monoclonals that they characterize, they are partially or completely escaped by changes present in at least some known Lassa virus sequences. So they say this shows how the development of antibodies should consider the potential for escape to avoid problems like we've had with SARS-CoV-2, where variants arise and then we have to stop using the antibodies. So I think it's a very cool study.
It's really
I like it very much. So now you have all the information you need. If you want to know how much can the GPC change, here it is. They changed every amino acid.
I'm looking at their interactive graphic of the mapping of the amino acids on the glycoprotein. It's pretty amazing. It's a lot of detail in a small package.
Exactly.
It's great. This is a public resource, so you can go look at it.
Very cool.
Yep.
Right. And I can't remember, you may have said it when I was looking at something else, that this allows them to have a BSL-2 method to look at this mutational space.
Right.
So like the first paper, the idea was let's get away from using, well, yeah. So this is the Lassa's BSL-4, and what they can do now is do some things at BSL-2. And the previous paper, SARS-CoV-2, is BSL-3, and now they can do things at BSL-2.
Right.
So both of them are getting to easier.
They're
safety
domesticating
levels.
these viruses.
Right.
Yes.
So I have a question for the general audience. What is the function of the receptor molecule on the cell surface when it interacts with its normal ligand?
This one I don't know.
The alpha-dystroglan.
I've never heard of that before.
Yeah,
That's a strange word for me.
alpha-dystroglan.
Yeah, that's the one.
That's the receptor.
Yeah.
I know.
But
Now,
he
what
wants to know
what is
what
it
its
already-
normal
what-
function
Right.
What's
What
its
does it
day job?
normally do?
What does it do?
Let's look.
Yeah, looking it up
Let's
now.
look.
It's there solely to bind Lassa virus.
Dr. Google will be consulted on this patient.
It is a cell surface molecule that links proteins of the extracellular matrix with the internal actin cytoskeletal machinery.
Good Lord.
So it's an anchor
It's an
that
anchor.
keeps cell sheets together, basically.
So wait a minute. How does the virus get access to it then?
Well, it's on the cell surface.
I
Yeah, it's
understand
not
that,
all.
but
It's not all
linked.
stuck to the
It's
matrix.
not all busy.
Okay. Well, okay. That's fair enough.
Let's do a couple of email here. Do I have a volunteer for the first one? It's rather long.
You do it.
No, I don't want to
I'll do it.
No,
It's all
Alan.
about you.
Go ahead, Alan.
All right. Fernando writes, "Hi, TWiV hosts. I'm a longtime listener, recommender, and financial supporter of the podcasts. I have a background in several areas of science and technology and even a few papers listed in PubMed. I enjoy and support what you do, but your, mostly Vincent with Dixon's support, rants about conflicts between science, politics, and law, and specifically about the Chevron doctrine, are losing the plot and risk alienating otherwise well-disposed listeners. Here's why. None of us who have worked in science and technology over the last 50 years can ignore that it's ever harder, even for good faith people, to understand and act wisely in an increasingly complex technological world. Whether it is the economy, environment, health, technology, even as many of the worldwide baselines have become objectively better, less poverty, more medical successes, less everyday pollution, easier everyday living, it's also become harder for even scientifically literate folks to manage the complex, opaque processes that our everyday lives depend on. The epitome of this were the many misguided policies of health authorities during the height of the COVID-19 pandemic, where substantial restrictions were imposed on people's lives and livelihoods with claims of scientific backing, when in fact, as your discussion in this same episode of the new WHO respiratory infection guidelines showed, much of the so-called science on infection control was medical just-so stories. It got to extremes like my local health authorities closing hiking trails and children's playgrounds for months because of alleged infection risk, not to mention the abusively long closing of public schools. So why are you shocked that political opportunists and grifters take advantage of understandable public lack of confidence in scientific opinion that hasn't exactly covered itself in glory? The defense you sometimes use, that there's much uncertainty in science and so science should be forgiven for later corrected errors, tastes very sour when those who claim scientific authority in their public policy decisions have shown no such humility. Sure, we are skeptical that judges will always make scientifically sound interpretations of federal laws. The long and successful attack against the Chevron doctrine was well funded by the obvious self-interested parties. But again, it's not as if federal agencies wrapping themselves in the magic cloak of science are exactly blameless here. For example, those of us in the American West who know folks who try to make a living from the land here learn about many arbitrary federal decisions in the name of scientific conservation that aren't anything but petty exercises in bureaucratic authority or careerist cowardice. I appreciate you already do a huge job of advancing science education through all of these podcasts, but your frustration with anti-science demagoguery should not make you unable to recognize that trust in science and scientists needs to be constantly earned. Blaming the messenger does not change the truth of the message. Okay.
All right. So I just want to say that when you say we're going to alienate people by expressing our opinions, I think that's very unfair. TWiV is all about expressing your opinion, as is the world, right? That's the way it's supposed to be. So I don't get this, you're going to alienate me. We can disagree. That's fine. But why do you have to leave if you don't agree with what I'm saying? I'm just upset that expertise is going to be ignored. That's the bottom line. I expressed my frustration with it. I understand everything that you say, Fernando. Science is not perfect, but when you want to make decisions, scientists need to be involved. Scientific decisions, scientists need to be involved. That's all I'm saying.
Vincent, I support you.
Well,
I took a wait and see attitude on this because I'm hoping that the expertise is not ignored. And I pointed out that there is phraseology in these rulings, leaving open the door
yeah.
for appropriate scientific input. And I think the point of the letter is that these are complex issues. And I think in the example of COVID is great because there's this really dynamic tension between the science and the social impact of the regulations. And we've talked before about my perspective on this is that a bunch of different things were tried. Probably nobody got it right. And I would love to see the dissertations that come out of this down the road. I hope they're being done to try and figure out what might have been an appropriate path to take, though it would have been hard to find that initially because we didn't know what the hell was going
But
on.
we did do some things right, like
We
make
did do some
vaccines
things
that
right.
bloody work.
Yes.
Right.
But one of the problems here is that there's mistrust of the science. There's also mistrust of the people, of the lawyers, if you like. And there needs to be an appropriate trust and balance between the two to make this work right. Okay, whether that's going to happen or not, I don't know. We'll find out.
I can also defend those people in public office who have to make a decision based on the immediate scientific results, like a Tony Fauci, for instance, who makes this decision based on the current data.
Yes.
And then as the experiment goes on and gets more data, and it changes the landscape, so to speak, of recommendations, you're allowed to change your mind.
Yeah, and I would like to look back and, there's the thing in here. Those who claim of scientific authority in their public policy decisions have shown no such humility. I wonder about that because
I disagree.
I've heard Tony say I'm wrong.
Yes.
That's
Or
right.
I was wrong. Okay.
Many times.
And I think he should not have been the face of the government response. The director of the CDC should have been. So he was thrust into this role. And I think he was actually very, very upfront about what he didn't know.
Yeah, this is what we know now.
But there was also a lot that he knew. And in large part, and he was not the primary mover behind a lot of these policies, by the way.
No, certainly not.
So a lot of what was done was erring on the side of caution. And
Right.
you can fault that and say, well, we shouldn't have erred on the side of caution. But I would disagree. I think in the case of a pandemic that's killing millions of people, you start out with the more restrictive, safer policy, and then you back things off as you learn more.
And the voice of reason that was missing in all of this was the Surgeon General.
Yes.
Who also has the responsibility of conveying science to the people and allowing them to digest, so to speak, what science has indicated is the right approach. Masking, distancing, vaccines, all of these things fall into that thing. But the Surgeon General at that point was not.
No, the Surgeon General and the director of the
forthcoming.
CDC at that point were not useful.
Neither
context
person
of-
was.
So-
So Tony stepped it up.
Here's another aspect of the problem. So we have two of the presidential candidates who are markedly anti-vaccine. There was a conversation between Trump and
That's right.
RFK Jr. recently released
That's
where the
right,
president said,
that's right.
these childhood vaccines are out of control. It's too much. It has to be changed. As if he has any knowledge of this. And he said, I want you, RFK Jr., to help me. I'm going to be elected and you're going to help me do that. So there's this specter that RFK Jr. will be head of CDC or HHS and completely screw everything up. That's what I'm talking about, Fernando. You say to me, I am unable to recognize that trust in science needs to be earned. Of course I recognize that. Why do you say something like that? I'm just railing against the loss of expertise. That's all.
And calm logic and reason over hyperbole and ranting. And Vincent doesn't
I
rant
do
much.
rant. I'm sorry, I
He,
rant,
he
but
rants sometimes.
Well, I said
he
much.
definitely
you
I
know,
said much.
most of my
When he
career,
does, he's usually right.
most of my career, I ranted to my colleagues who were around me in my office at Columbia.
Yes, you
right?
did. Yes,
That's
you
what
did.
we do. And suddenly I have
Yes,
this
that's what we
platform
do.
where people wanted to hear what I thought and us about what we think about things. So that's what we're doing. And you may not agree with it, but this is our opinion. And I hope to learn all the time from this. But
I think
Thank you
it's
for your
important.
input.
yes,
I think it's
Yes.
important to have open dialogue and discourse and hear opinions from all sides, and that's what this is
Kathy,
all
you're
enabling.
absolutely, and this letter is
Yes.
proof of that. We
Yeah.
read the letter,
Mhm.
and now we're responding to it.
Mhm.
Dixon, can you take the next one?
Sure. Suzanne writes, while listening to Alan's recommendation in episode 1129, I thought he might enjoy this book that was recently added to Project Gutenberg. I haven't finished reading it yet, although it's relatively short, but the beginning is delightful. A father, a son, and their cat sail from the northeast coast down to Florida a little over 100 years ago. The father made a book out of his log.
This is
Take
a
it,
fun
Rich.
book. I should clarify, the cat in this case is not a feline. It's the catboat that they're sailing, which is a specific type of sailing rig.
Is it a catamaran by any chance?
No, it's a catboat is different from a catamaran. I know
catboat's
this is
different,
confusing. A catboat is a monohull boat that has the mast very far forward and a single large mainsail. That's the catboat rig, which was a standard working craft in the United States from way, way back into the into the 20th century, and this is from the early 20th century, 1912 to 13. I have read it. This is a fun book. It's fun in a lot of ways. I mean, the sailing parts are neat, but also this is America. This is the coast of the United States, the east coast of the United States in 1912. And it is really different from everything. I mean, we didn't have the intracoastal waterway. We didn't have a whole bunch of beachfront luxury homes all along these areas. And this guy and his kid are just sailing down the coast. And they're, in a lot of cases, they're foraging for their food along the way. And I should point out, I think this is revealed at some point in the book or revealed afterwards, or it's just implied by context. Plummer frequently references, this was the log from the boat, and he frequently references they went ashore and got a couple of pheasants and had them for dinner. And reading along, I thought, man, there must have been a lot of pheasants, or these guys are really successful hunters. And you gradually figure out when he says pheasant, it's code for they stole a couple of chickens.
Right.
So they went ashore and raided somebody's hen house, and that was their dinner.
So these books are free, and if you just click on the link, you see a picture of what that kind of cat boat is. But these books are available for Kindle, older Kindles, all kinds of e-readers and things. So
my understanding is that the original printed bound version of this was the guy, I don't know where he got it printed, but he hand bound it with fishing line.
Cool.
And released it. But the obviously when you download it on your Kindle, you don't get that effect. And I did not. I read it digitally as well.
Yeah. Rich, can you take the next one?
Sure. I took the one before
let's see here. James Wright. James Wright. Vincent. And starts up with a link to a Nature article entitled If Bird Flu Sparks a Pandemic, Your Past Immunity Could Help. Wait a minute. Okay, fine. This Nature article is very intriguing. The 2009 swine flu pandemic. I received the vaccine for this and wonder if it will work as well. That doesn't leave people completely unprotected because exposure to an older pandemic flu strain can defend against a newer one, says Michael Warraby, an evolutionary biologist at the University of Arizona, Tucson. For example, in a 2009 pandemic caused by swine flu virus H1N1, 80% of deaths were in people younger than 65, older generations were spared owing to immunity stemming from the exposure to different H1N1 strains when they were younger. Yeah, we discussed that briefly last time. Exposure to H1N1 during a 2009 pandemic and at other times might in turn provide some protection against H5N1 strain on the rise today. Both the H5 and the H5N1 and the H1N1 viruses have a surface protein designated N1 and an immune system that responds to H1N1 might also respond to H5N1. Pyrrhus and his colleagues found that the near universal exposure to H1N1 in 2009 and subsequent years produces antibodies that respond to H5N1 in nearly 97% of the samples they collected. He is now running animal experiments to determine whether this antibody response confers protection against infection and serious illness. Keep up the great work, James. Right. So past flu exposures, even to things that on the surface seem unrelated, okay, could provide protection against what it seemed like novel flu and influenza viruses. Thank you.
So he asks if his 2009 vaccine will protect him against H5N1. Well, that's what Paris is doing to figure out whether that's good enough for that. But that's natural infection, so I don't know about vaccination. Could be different. But yeah, this is an interesting article that we've talked about before. Kathy, can you take the next one? Peter.
My mouse just stopped working. I'll go as far as I can. Peter writes, I really appreciated your recent detailed analysis of the claim that the COVID virus originated in a lab, but there's one part of your analysis that I don't think you addressed. Forgive me if I missed or misunderstood something in the analysis. You state that the COVID virus was so drastically different and in such an unusual manner that it would not have been designed that way in a lab, and thus it could only have originated randomly in nature when it appeared in the animal market. But I would think the same argument could work the other way, that the variant was so different that it would be unexpected that it would suddenly turn up naturally in the market without any more subtle variants appearing in the market or in other places. For example, maybe variants that make people mildly ill versus seriously ill. What if the variant we saw in the market was a variant from a lab that accidentally got loose? Thus, it wasn't designed that way, but was simply an errant version of different things they were testing that got out into the wild. Given the poor containment reputation of the lab, it would seem like a plausible scenario. I realize testing was probably better around Wuhan, but if such a variant occurred naturally, wouldn't similar variations have been recorded eventually in other geographies? I would appreciate your perspective on this. I am not always able to listen to all of every episode, so if you respond on your podcast, I'd appreciate it if you would let me know which one. Thanks for such a thorough analysis of the topic. Is that at the end of it? Because I can't scroll up.
That's it.
That is the end of it.
Good. Perfect. So I was so trying to fiddle with my mouse, I wasn't really getting into the content. So you guys can jump in, except that the statement of, given the poor containment reputation of the lab, I know of no evidence.
Citation needed.
That there's was any evidence for poor containment at the Wuhan Institute for Virology.
So the problem is, Peter, that if you're saying it got out of the lab, then it would have to have been there, and there's no evidence that that lab had SARS-CoV-2 or anything close to it. They had SARS-1-like viruses for sure, but they're too distant to have become SARS-CoV-2. So what you're positing is that it was a variant from a lab that got accidentally loose. No, it can't be a variant from the lab because no one had that virus.
And importantly, no one had anything like that virus in the lab or anywhere else. There's no evidence supporting that notion at all.
What I find... Go ahead.
I'd like to riff on the first part of this, that you state that the COVID virus was so drastically different and in such an unusual manner that it would not have been designed that way in a lab. Okay. Um, Thus, it could only have occurred randomly in nature. I would think the same argument could work the other way, that the variant was so different that it would be unexpected. It would suddenly turn up. No. Variation, first of all, we only know the minute fraction of what's out there in nature, and variation in nature is boundless. Okay, there we can anything is possible, and it's happening all the time. The deities are testing us constantly. Okay, with variance. So there's an infinite possibility in nature for variation.
And I just want to address this notion that we would expect some kind of progression of the viral isolates within the initial isolates, like there should have been something that showed this evolutionary change. That's not how a spillover works. A spillover is whatever variant was capable of infecting humans. And by definition, in this case, we're only going to see a variant that could not only infect a human, but could transmit to another human. So it has, once it gets to that point, it has already cleared this huge hurdle. And no, I wouldn't expect a lot of changes or multiple variants in that. I mean, there are actually two clades, but they're very, very similar in the overall structure of the virus because they both had to clear the same evolutionary hurdle. This is exactly what we expect and exactly what we see in well-documented spillovers.
All right, one more from Rena. Hi, all. Love your podcast. Question. Historically speaking, do viruses that spill over from animal to human immediately have the capability to go human to human right out of the gate? Or does that take time for a natural-born virus? You also have to assume that the government isn't lying, in particular the Chinese government, which does not have a reliable history of truth-telling. All right, so the first question, it can be either. The problem is, by the time we find something in people, we don't have the original. Now, SARS-1, we have some isolates from animals, but that we don't have those until 6 months after the outbreak began in people, right? And we can see that the animal isolates are different from people. So the assumption is maybe they underwent some change, but the timing isn't right there. We think maybe for 1918, the virus came fully baked out of a bird, ready to infect people, but we don't have any data that we can use to support that because there are no isolates except one of the 1918 viruses itself. So I think both are possible. Sometimes just randomly in some animal in nature, a virus will emerge that is ready to go in humans. And if it contacts a human, it may be ready. Or
not just ready to infect a single human, but to transmit
right.
from human to human, which is really what Rena's asking.
That's right.
And we actually have, I mean, the view that people have come to realize in the past few years is spillovers from animal to human that do not have the capability to spread human to human are a lot more common than we realized.
Right. Yeah.
And the reason we didn't see them before is because they don't have the ability to spread from human to human. We just talked about a virus today, Lassa virus, that is almost exclusively seen in animal to human spillovers that don't go to another human.
Right.
Thousands of people die of this every year because the animal happens to be a peridomestic rodent pest that people have a lot of contact with. But this notion that there are constantly spillovers happening, they just don't gain traction to spread from person to person, and we don't see them because in many cases, either the original infectee doesn't develop symptoms, or they develop symptoms that are so bad that they die and nobody diagnoses what happened. So in order to have a spillover that we actually notice, it almost has to be one, unless it's from a very common animal, it almost has to be one that can spread human to human. So again, the barrier to observe this is so high that the examples we see, such as SARS-CoV-2, are the ones that can spread human to human. Even if that's less common, that's just what we happen to be able to see.
Now, with HIV, we know that it spilled over from chimpanzees or gorillas. It was SIV, and that's quite different from HIV. And maybe if it spilled over in 1920, we didn't recognize it until the 1980s. So maybe it took a long time for it to undergo the changes that allow it to transmit among humans. So I think both work, and we don't have a lot of data in either case.
The example that comes to my mind is the old hantavirus story from the Four Corners in the U.S. It must be 30 years old now, at least.
Yeah.
Where this is a virus that lives in rodents and under certain environmental conditions. It's shed in the rodent urine under certain environmental conditions. Rodent infested spaces, when they're dried out, kick up a lot of dust that has virus in it. Humans can catch it and they get a respiratory infection. It's not transmitted from human to human, but can show up under the appropriate environmental conditions as an outbreak of respiratory disease. Now, interestingly, there's and actually, once you look into this, you find, oh my God, there's hantaviruses all over the place, okay, that are doing this all the time. But as Alan points out, we just never knew about it because they don't spread all that rapidly. But there are a whole bunch of respiratory infections out there, the etiology of which is not known, okay, but they may be similar kinds of things. And a lot of these viruses that spill over cause similar symptoms. So you can't, somebody shows up with a respiratory infection, you don't know what the agent was. Could be one of these spillovers.
All right. Thank you for your questions, folks.
And just in terms of the second part of the letter, we don't have to assume that the government isn't lying, because the evidence that they would have to lie about is so extensive that it extends to things we can verify.
Yeah, in fact, most of the data that we have supporting a natural origin is independent of China lying or not lying, right?
Yeah.
Right.
In fact, they published the environmental sequences from the market, and those support the, the idea that it was the epicenter. Yeah. Okay, let's do some picks. Dixon, what's up this week?
Well, a little space mining. We've got 2 rovers still active on the surface of Mars, and they both have capacity for digging and finding. And one of them cracked open a rock, and out came elemental sulfur.
Well, that stinks.
Yeah. Actually, it doesn't stink.
No, it doesn't.
Have you ever been as close to a cake of sulfur that's as high as the room I'm sitting in and twice as long? And I have.
No.
It's in Alberta. And it's the gas, hydrogen sulfide, which does stink. And they mine the sulfur, and the sulfur is shipped over to Japan, and Japan makes very, very pure versions of sulfuric acid with it.
Mm.
So, but to find elemental anything on Mars is quite remarkable.
That's pretty cool.
And very cool. And it suggests that there's a sulfur cycle. And sulfur cycles typically are volcanic, but also in the on Earth, they involve this little algae which takes the sulfur from the black smokers and creates dimethyl sulfide with it and throws it back in the atmosphere. So it's quite an interesting finding. I thought it was the first remarkable finding that those rovers have actually found.
Excellent.
Very cool.
Kathy, what do you have for us?
I picked something that I've been waiting for. It's now up on the ASV website. So the Lynette lectures at ASV are something that Rich Condit kind of pulled out from being on the History and Archives Committee. I think that their purpose was to have someone talk about the history of virology through their own particular career. And that had kind of drifted away from what the Lynette lectures were, but the last 4 have been that way, and all 4 of them are now on the website. Kay Holmes' lecture has been on for a while because it was a virtual one in 2021, And now we also have Lynn Enquist, Roz Sandry Golden, and then this year's from ASV 2024 was Gail Wirtz. And her lecture was amazing. And I just want to tell you a couple of the highlights. Each of these is only about a half hour long, and I'm recommending that Gail's could be used in teaching in a virology class. So she was a student with Julie Youngner, and then she ended up being at University of Alabama, Birmingham, UAB, and then at UVA, and then she finished up at College of William and Mary, which I think had been her alma mater, and she went there for a master's degree in 2020 in archaeology. So just really, really great. But she started off the lecture talking about Julie Youngner and his work on the polio virus, and he and two other people were very instrumental in the work of the Salk vaccine. Those other two people were James Lewis and Byron Bennett, and they and also Percival Baisley, she mentioned. And Salk did not acknowledge one of them in his announcement in Rackham in April 1955. Anyway, so then Gail went on to talk about her own career and how various mentors inspired her, and part of her career. Rackham, of course, was here at the University of Michigan. So then her other University of Michigan connection was that she did her postdoc with Mike Levine. And so when she was there, he went on sabbatical and she taught his class. But she also said that there were 3 women in human genetics at the time, and 2 of them were in Mike's lab. So it was a good environment for her. And she also mentioned that she worked at the bench throughout her career, and her excitement was in carrying out her own research. And I could really relate to that. So anyway, it's just a fascinating lecture. I highly recommend it. The other 3 have also been very good. So you get there by going to the History and Archives section on the ASV website. Scroll down a little ways till you see the Lynette lectures, and there you are.
Cool. I'm glad to see those being used for their intended
Yes.
purpose. That's
Yes.
good.
Yeah.
Rich, what do you have for us?
So this came to me from my son, and I need to introduce it by introducing what my son does. And I had to have him give me a 25 words or less elevator pitch so that I could describe it. And I commented to him, it's really cool when your kids are grown up enough so that you can't understand what they do. He works for Google, and he's very interested in doing what he can in the arena of climate change. And he says, quote, I work on Google Cloud Carbon Footprint. Google Cloud serves business users who want to use Google's data centers to power their businesses. And data centers are energy intensive enterprises. We report back to them, that is the users, on how much carbon we've emitted as a result of their usage. Okay. And my pick is a thing called electricity maps, which I'll describe. But Jeremy says, the data from electricity maps is used to determine how much carbon is emitted per kilowatt hour of energy consumed at our data centers. So they describe energy maps as a partner in this enterprise, okay? And they use it to help them understand the carbon footprint at various centers. So if you look at this, I've included the link. It is a world map divided up into various segments and colored to indicate the carbon footprint of any given region. And you can hover over any given region, and it will tell you a quantum called carbon intensity and how much of their energy is from low carbon or renewable resources, etc. So you can look over your own. And if you click on a given area, it will actually break it down into various resources: geothermal, biomass, solar, wind, nuclear, etc. So you can, zoom in and out. You can look at various countries or your whole area. And when we were having this conversation, just to elaborate a little more, he pointed out to me, if you look at Europe,
Mm.
It has a variety of colors, but France is deep, dark green.
Yes.
Okay. And it stands out from the rest of the European continent.
Yep.
And he pointed out to me that that's because they nuke most of their energy. Okay.
Yes.
Which brought up the issue of, okay,
Mm.
in this era where we're trying to reduce our carbon footprint, how about nukes? And so he brought to my attention a couple of recent opinion articles from the New York Times, which I've also linked, that I'll just say bat around the pros and cons of nuclear energy, mostly in terms of the problem that's usually associated with it is nuclear waste. I think the safety issues, well, not completely. There is Fukushima out there, but the safety issues can be addressed. The waste issue is, it's an issue. Okay,
It's
but
thorny.
It's thorny. Well said. So, but it depends to some on your extent. Depends to some extent on how you look at it, how thorny it actually is. At any rate, the energy map is fun. The problem is interesting. So play with it. I'm looking at Poland.
Very,
here. Man,
very cool visualization.
What's going on in
this.
Poland and South Africa, man? They're all the way at the end of
Poland
the
is
sphere.
like mostly coal.
Coal.
That's
Yeah.
correct. That is correct.
In South Africa
And there,
too?
there are some significant gaps. I mean,
Yes,
most
South
of
Africa
Africa is
too.
missing. A lot of South and Latin America are missing. China,
Yep.
missing data. Some states in the U.S. missing data. So
I wonder
I'm going
how
to follow
China...
up. I'm going to follow up on this later on because he also with more on Google's project, because
Yeah.
Jeremy sent me a video with more detail about exactly what they do, and it's really interesting. I mean, they're thinking about stuff that would never cross my mind, and about how you can fiddle your usage in terms of what data centers you use globally
Mm.
and when you use them to minimize your carbon footprint, etc. Okay.
So run your cursor over Iceland, and you'll get
yes,
100% renewable.
because it's all
And no
geothermal.
nuclear. No nuclear.
It's
And
all
if you do that to
geothermal.
Sweden, yeah, well, that's not nuclear.
No, right. I mean,
Sweden's
that's
not
why
nuclear.
Iceland is uniquely placed because they're sitting on top of
No,
a
I've
whole
been
bunch
to
of
Iceland. I
hydrothermal
understand
vents.
that, but take a look at Sweden, for instance. That's mostly hydropower.
Yeah. If
So
you
it
have
depends on where
the
you live.
advantage, if you have the advantage of being blessed with a location that has a lot of
Exactly.
renewable energy, then that's easy to do.
Exactly.
Uruguay is really good.
Right.
So parts of Brazil also.
Yeah.
No part of the U.S. Well, I guess the Northwest is green.
Missouri, the state of Missouri is just almost black.
No, the
There is a
Washington.
section of South Carolina that is.
Yeah, Washington does pretty well. Parts of Oregon, but
Well,
boy, here
yeah.
in the East, forget it.
Well,
And you know what, Dixon? It's going to get worse in the next 4 years.
Probably.
Well,
We'll see.
I hope you're wrong, but
I hope so too.
I tend to agree with you most of the time. Yeah, it's
Thank you.
it's
That's
a
cool.
sad
Thank you, Rich.
situation.
That's great.
I love it.
Alan, what do you have for us?
I have an article, a news article from Nature. They sent a reporter. They did a little investigative report. So I'm sure we have all gotten this endless barrage of spam.
This is good.
From the conference, the International Conference of blah, 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 that you've never heard of, and come present your prestigious data. That I get these, and I have not published an original research paper in over 25 years, so everybody who has done any kind of lab research gets these emails. And I always kind of wondered, do these conferences, I mean, what happens if somebody signs up? Do they just is it just a straight up scam, or is there actually some kind of meeting going on that is just part of it? It turns out, at least for one of the big players in this, they actually host something that could be called conferences. And the suckers, I mean, the individuals who click on the link and register for the conference, and go to one of these, do experience some sort of meeting in many cases, but it's not what they had signed up for. And this article is really good in a lot of ways. So they sent a reporter to one of these who it described it as a shambolic experience. It's just it's a small group of people in a hotel conference room who nobody's quite sure what the conference is about, including the organizer who was who was brought in to do this on, as part of the scam, basically. They don't even really know. They're just a postdoc who clicked on this. And so it's a useless conference, but the article points out that the participants are not necessarily blameless either, because in many cases they are on the treadmill of having to present at international conferences in order to get tenure, in order to continue to have jobs. And so they're desperate to find a place to present. The big conferences in their field often don't have any space for them. And so they go to these predatory conferences to be able to say, I attended this international conference of blah, blah, blah in London, and put that line on their CV. And, the companies doing this are making massive amounts of money off it. So it's not going to stop anytime soon. But it's interesting to see what these actually entail.
Very cool. I like this.
Can you imagine? Oh my gosh, it's horrible.
Yeah, and a lot of the people they talk to people, some of whom don't want to be named, but they don't manage to get any reply from this company called Omics. That's one of the big purveyors of this sort of nonsense. But they do talk to people who have attended these conferences, and some of them are very upset, obviously, about having flown halfway around the world in some cases to go to something that turns out to be a complete waste of time.
So the illustration for this is amazing. It's a picture of a wolf, and its top teeth are just regular teeth. And the bottom teeth are a set of chairs lined up in an auditorium and a podium. It's just really creative.
Illustration by Sebastian Thibault, who I think has done a bang-up job on that.
I have a similar gripe about these fake universities, like University of the Grand Canyon, and there's a couple of others. There's one that has a football stadium in, I believe it's in Phoenix, and they claim that they've got, presence because look, they host the such and such bowl. I forget which bowl it is, but it's a football conference after the regular season, and they've actually had pro football games held there as well, and that's their presence. They have no buildings. They have a faculty that's on paper only, and you can take your courses online, of course, and get your degree sent to you in a very, very fast period of time, depending on how much you pay.
Yep.
Wow.
Why do we allow that?
A lot of things happen in the world, Dixon.
Who's regulating that?
Nobody.
You'll remember that Mr. Trump had his own university
a lot of these
also.
Yeah, a lot of these are driving through various types of loopholes, like the conferences, and people who feel like they've been ripped off are hesitant to report it because they don't want to admit to having been suckers.
It's absolutely horrible.
Oh, it's like predatory journals, right? They want you to submit, and you don't get anything out of it.
I'm thinking of safecrackers, right, Vincent? I mean, Dixon. For every phenomenon, there's going to be somebody trying to cheat the phenomenon.
Every successful system
never going to
draws parasites.
That is absolutely right.
Successful systems attract parasites.
But also, I mean, a lot of this, in fact, most of it maybe, is a byproduct of these systems where, we've gamified science so that people are promoted and tenured based on numbers, based on citations, and again, a lot of these people going to these conferences are told you have to attend 3 international conferences or you're out of here.
Right.
And so this creates the need for this type of thing.
Yuck.
So the system is what's broken fundamentally, and then some people are taking advantage of that.
Okay, speaking of broken, and also speaking of coal. I have an article in Ars Technica by Barbara Pinot. Coal-filled trains are likely sending people to the hospital. coal-filled trains trail a cloud of particulates shaken free from their cargo. So this is an article about a very interesting study published in Environmental Research, where they monitored particulates coming off of coal trains. The U.S., still uses millions of tons a year, and we transport it in trains. And the study found that people living near railroad tracks have higher premature mortality rates due to air pollutants released during the passage of uncovered coal trains. They measured the particulates coming off the coal trains. They did San Francisco, Oakland, Richmond, and Berkeley. Increases in air pollutants, such as small particulate matter, are also associated with increases in asthma-related episodes and hospital admissions. So you should read the article because it's pretty amazing. Like people in Richmond who live around the rail line have mortality rates 10 to 50% higher than the county average.
Wow.
And
that's amazing.
I mean, a low-tech solution would be to cover the coal.
Cover the rail cars.
But it costs money.
Absolutely.
It costs money, and at the scale, a lot of people don't appreciate the scale at which this operates.
Yeah.
I mean, you mentioned millions of tons, but to see one of these trains go by, if you're ever stuck at a crossing when a coal train is coming, you're going to be there for a while.
That's right.
And it's just, these things can be a mile long, and each car in that train is multiple tractor trailer loads of coal.
Yep.
Covering all of those hundreds of cars is going to require not only the material, but the labor. And then you'd have to uncover them at the destination, and that is itself a problem because the way these trains pull into the power plant, and they actually have a tipper that grips the car and turns it over and dumps it out like a giant cup, and then flips it back and then goes to the next one and dumps it over, and you'd have to then remove the cover. And the reason we're burning coal is because it's cheap.
Yep.
Kathy, you're muted.
You're muted.
You're muted, Kathy.
You're still muted.
You're still muted.
She knows she's still muted.
Okay. The cover could be designed such that it would open up as the car flipped. So that's not a problem, but they do say that covering the coal cars could increase the risk of fires.
Yes.
So that's another issue.
So
there's always an excuse.
I imagine the people working at the plants where the coal is dumped, it must be even worse.
The coal mines.
Well, that's actually pretty well documented. Black lung is an occupational hazard for anybody who works with coal.
So, you can imagine that certain people in the U.S. would like to see more coal produced, and they don't care about this. They're going to find ways to discount this study and say, no, it doesn't matter. But it's another example of how we do things and don't worry about the impact on people's health.
That's
It's really unfortunate. Anyway, it's really an interesting study. It's a very, very, it's never been done before, apparently, and the way they designed it is quite interesting.
And it's a good article. It's well written.
All right, that's TWiV 1133. You can find the show notes at microbe.tv/TWiV. If you have a question or a comment, you want to send us a pic, TWiV@microbe.tv. And if you like our work on this and our other programs, please consider supporting us, microbe.tv/contribute. Dixon Despommier, trichinella.org, thelivingliver.org. Thank you, Dixon.
You're welcome, Vincent.
Rich Condit.
I had a great time.
You had a good time, Dixon? I'm very happy to hear that.
I had a wonderful time.
Now you're going to go out and eat on your terrace, right?
We are.
Very good. Enjoy.
Some kind of a shrimp dish. My wife popped in while I was on air and told me that's what's on the menu for
All right,
tonight.
I'll be there.
You're invited. Absolutely. All of you. Come on.
It would take me a while.
We'll wait.
No, that's all right.
It would take Rich the longest, I guess, right?
That's right.
I'm not coming.
Rich Condit.
He's allergic to
He's an
shrimp.
emeritus professor, University of Florida, Gainesville. He's currently in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time.
Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
You're welcome. It's fun to be here. Thanks.
Alan Dove is at alandove.com, turbidblack.com. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV, Ronald Jenkins for the music, and Jolene for the timestamps. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.